So it's my great pleasure to introduce today's speaker, Dr. Rich Boden. Rich is an associate professor of microbial physiology and taxonomy in the School of Biological and Marine Sciences at the University of Plymouth. And he is also one of the trustees of the Linnean Society. So he's joined the council of LINSOC just over a year ago. And he's already been an absolutely massive contributor to many of the initiatives and changes that we have going on in the Linnean Society, really making sure our society is as impactful and effective as possible for the membership and for our broader society. So thank you, first of all, Rich, for all of your great service. Now, Rich has a really interesting field of research that really spans both applied and basic science. So most of us are usually on one or the other side, but he really spans both of these fields, looking at everything from the physiology, ecology, biochemistry, taxonomy, and environmental processes of soil and freshwater, including soil and freshwater geochemistry, and really concerning the microbial transformations of metals, metalloids, and sulfur in a range of environments. And so this touches on a vast range of topics in the environmental sciences, is obviously of huge interest as we think about the world that we live in today and the changes that we're going through in our environment. In addition to all of this exciting scientific work, and of course, all of his work with the Linnean Society, he's also the editor-in-chief of the FEM's Microbiology Letters Journal, which is an absolutely massive journal with a huge readership. And of course, he does all this in his spare time, like many of us do. So it's really, really amazing to see all his various accomplishments and all the work that he's able to do in leading in very many aspects of his field. So with that very, I think, superficial introduction, I'm really looking forward to hearing more about his work today with his talk titled New Insights into the Thiotrichales, including hydrothermal vent species. And I probably didn't say that correctly even after practicing, but hopefully that close. Thank you, Rich.
Okay, I'm well aware from having done previous talks at the Society that a lot of our membership don't know a great deal of microbiology, so I've decided to do a gentle introduction up to the point where I cover what I want to cover, which is on this quite diverse order, which is probably really 3 orders. We're working on that at the moment, which includes all sorts of environments, including the ones that I've shown on this title slide, everything from soda lakes, volcanic lakes, gills of clams, through to deep sea vents, and also a lot of benign environments like soils as well. I'm going to do acknowledgements at the beginning because otherwise I always forget to do them at the end. And I really want to acknowledge 2 of my former project student and an intern, Rory and Alex, who did a huge amount of the work in the second part of this talk. And then the carbon uptake work that I want to talk about, which was pioneered by Katie Scott at South Florida, and I tagged on really to do a very small piece of that, but I want to tell part of that story because it is quite interesting in our general understanding of these organisms, and it relates to vents, which I think most people find nominally quite interesting. So these types of organisms are quite different from a lot of things that we tend to see and think about. They're very similar to plants without the light, effectively. So if we look at the diagram here of what plants and algae do, ultimately what they're doing is taking water, which is their electron donor, splitting it with energy from light, which gives off molecular oxygen, as we know. And what it also gives off is electrons. And some of those electrons are harnessed on a carrier, which is NADPH, and the remainder are used to make ATP, ultimately. And that NADPH and that ATP are used to convert carbon dioxide into biological material, into cells. And that whole process we stick together and call photosynthesis. In this particular group of bacteria, which are very diverse, they're found all over the world in all sorts of habitats, it's chemosynthesis. It's the same idea in the absence of light. And the reason for that is that these reactions, like water to oxygen, are thermodynamically impossible unless you've got some extra energy coming from somewhere, namely light. These reactions that bacteria do are actually thermodynamically favorable, so light is just not needed. And they take things like hydrogen sulfide and produce sulfate, and in the process take electrons make the same electron carrier and ATP and use those to fix carbon dioxide into biomass. And they actually do that in the same way as plants. They actually do that using the Calvin cycle. There are bacteria that use 5 other ways of taking up carbon dioxide, but the ones I'm dealing with today use the Calvin cycle. So we refer to them as chemolithoautotrophic bacteria. Plants are photolithoautotrophic. Carbon dioxide as the carbon source is autotrophic, self-feeding. Litho refers to an inorganic donor, water being inorganic, hydrogen sulfide being inorganic. And the chemo at the beginning is just where they're getting their energy from to make ATP, which is obviously chemical oxidation reactions rather than from sunlight. And some of the organisms I'm going to be visiting today are on this slide, because unfortunately they're not very photogenic organisms. On plates, they're just little white colonies. Under the microscope, they're a lot more interesting. So we've got Thiotrichales nivea on the left, which tends to live in sulfidic groundwaters all over the world. I'm not going to talk a great deal about that today, but Thiotrichales is what gives the Thiotrichales its name. It's named for that genus. And then I've got 3 species in the genus Thiomicrospira here, just to show the diversity of the shapes and the morphology, which is going to become important a bit later. I'm going to talk quite a bit about Thiomicrospira Thyasira, which comes from gill tissue of clams, Thiomicrospira cyclica, which comes from Mono Lake in California, or I think it's supposed to be Mono Lake in California, and Thiomicrospira pelophylla from the Waddenzee in the Netherlands, which was the first Thiomicrospira to be cultivated back in the 70s. So stepping back very far up to top level of life, because I think this is the bit where a lot of people don't even have this bit of the understanding. So I thought I'll start really zoomed out and then I'll zoom in. Life these days has got 3 domains to it. Many years ago, we had prokaryotes and eukaryotes, and we had eubacteria and archaeobacteria. All that's gone now. We understand life as 3 domains. There's the bacteria, which are shown here in violet, the archaea, which are shown in orange, and the eukarya, which are shown in green. And you can see the eukarya is the least diverse of the 3 clades, and that often surprises people. But the reason for that is that all eukarya have one common ancestor, and our mitochondria have one common ancestor, and chloroplasts have one common ancestor. So the actual metabolic capability isn't vast. We're all very, very similar in core metabolism, whereas the bacteria evolved a lot longer ago and diversified very quickly right at the beginning. So you have this much bigger display of diversity, and that's why they can do all sorts of different things. And it's shown on here that, as you can see, the eukarya really are an outbranching from the archaea. And we do know a very close relative of our common ancestor, which is the Candidatus genus Prometheoarchaeum. And what happened is a very close relative of it took up one of the Rickettsiaceae from the bacteria, and that evolved into mitochondria, and that gave rise to the eukarya. And then later on, various cyanobacteria, which are up here at the top, got taken up, and that gave rise to plants and algae. So that's ultimately how all of life relates. And the bit that we're going to be looking at today is in the phylum Pseudomonadota, which is this group of branches off the tree over here. And you can probably make out these are kind of the furthest away from the root of the bacteria. So these are really quite evolved bacteria, quite divergent bacteria. So I'm going to do a really, really quick crash course in taxonomy and systematics, just because a lot of you have interest in botany and zoology, and the way we do taxonomy is not quite the same. To start with, we don't use all the ranks that the botanists and the zoologists use. We don't use infraorder, we don't use subclass and all those things. We just have domain, phylum, class, order, family, genus, species, and occasionally we'll use subspecies as well. And I've given an example of that here, just so you can see an example of how the names work out. And hopefully the colors are visible to people. I did run them through a color blindness thing to make sure that people would be able to see them. But until very recently, we didn't have phylum as a formal rank. That actually only appeared formally in the last 12 months. So the phylum Pseudomonadota used to be called the Proteobacteria, written in inverted commas because it was never a validly published name. And our names, unlike botany and zoology, are what are called registered names, in that they have to all go through one journal, either through publication in that journal directly, or they're published elsewhere, and then that journal has to then agree that the name has been done correctly and put it on one of their lists. And if you don't do that, it's not a proper name and it's a nickname forever. And we did that back in 1980 to avoid ending up with loads and loads of synonyms for the same thing. And it's been pretty successful, but it does always shock the botanists particularly that we're so militant about it. But it's for a reason, and it does work. So taking the Pseudomonadota, which I showed in that previous tree circled in red, I've expanded them out here in a different tree. And I'm going to explain what these trees really mean in a moment. But all you need to know in these trees is that it just shows distance between things. And in this case, each of those triangles is hundreds and hundreds and hundreds of species collapsed into a triangle just so you can't see the detail and don't get bogged down. Now, when I drew this, which was in the end of 2016, everything on this tree, apart from the Aquificota at the bottom, were considered in the old phylum Proteobacteria. What we now consider to be the Pseudomonadota is just this top section. And I think quite soon the alpha Proteobacteria are going to be removed from it because they don't really fit. So I think they're going to disappear. And because I know people don't have a great understanding of, well, what are these and where are they? I'm going to do a really, really quick whistle stop through them. So the alpha Proteobacteria at the top is very, very diverse. You find things in there like Rhizobium, which is what produces root nodules on the Fabaceae. And these are the organisms that fix nitrogen gas into ammonia for the plant, otherwise the plant wouldn't grow. Things like Hyphomicrobium with these long prostheca that it has, which is found in wastewater treatment plants and is important in the cycling of nitrate and converting it back into nitrogen gas. And then things like Acetobacter that produce these veils that we call mother of vinegar, and it's used industrially to make vinegar from waste wine because it can ferment ethanol into acetic acid. The beta Proteobacteria is the next of the big triangles here, which is very diverse again. Things like Neisseria, which is a pathogen that causes meningitis and gonorrhea. Zoogleuia, which produces these quite large amoeba-like multicellular masses that can move about. And that's also important in wastewater treatment as it causes flock formation, which is part of the process. And then very rare organisms like Anwoodia, which has only ever been found in a few thermal springs around the world, one of which is the Roman baths at Bath, and that's a genus that I discovered, well, the genus that I named. The actual strain was discovered by Anne Wood, who we named it after years later. Then there's the Hydrothermal, which you can't see, but that's a tiny little triangle just above the beta Proteobacteria, and it's only got 3 genera in it, of which I'm just showing 2 here. These are obligate thermophiles that use hydrogen gas as their energy source instead of hydrogen sulfide, as I showed at the beginning. And one of these has interestingly been found in the ice just above Lake Vostok in the Antarctic, which is causing a lot of controversy about why it might be there and whether it's just contamination at the moment. And then finally, the Acidithiobacillia, which is this group down the bottom here, which is again only 2 genera, one of which, Acidithiobacillus, is used industrially for leaching of copper in leach heaps at mines all over South America. And these will grow at pH -1 very comfortably. And yes, you can have minus pH. And they use iron as their energy source and produce iron hydroxide, which is the brown precipitate you can see around the colonies on the agar plate. And then its more benign cousin, Thermithiobacillus, which is another Roman baths isolate. And there's only 2 species of that known, one of which from the Roman baths and one from rotting lead ore in a museum somewhere in the Far East. Now, what we're going to be looking at today is the Gammaproteobacteria, the big red triangle. And even though we're looking at the Thiotrichales that live in hydrothermal vents and they're autotrophic, there's a great diversity in this class as well. Escherichia, you all know Escherichia coli, which is a very common model organism, and it's also a gut pathogen. Methylococcus, which you've probably not heard of, which lives in thermal springs, uses methane as its carbon source, and obviously therefore is really important in greenhouse gas control. And things like Xanthomonas, which is a plant pathogen that causes black rot, but it's also used industrially to make xanthan gum, which has become increasingly important with the proliferation of gluten-free food, as you can use the gum to replace the elastic properties of gluten, and you can buy that in supermarkets now. So within the Gammaproteobacteria is where we're going to delve. And just to give a quick aside on what the trees actually mean, all you need to know about them is the length of the line between any 2 names of organisms gives you a level of relationship. The longer the line, the less closely related they are. Now, these trees are strictly drawn on the basis of sequences. They're either drawn on DNA sequence or protein sequence. So what you're seeing is not literally the relatedness between 2 species, but between the same gene in each of those 2 species. And it's a good proxy, but it's not exactly the same as relatedness. And if you did trees with lots of different genes or lots of different proteins, you will see differences. But there are certain genes and certain proteins that we know work very well for showing differences, namely the ones that make up the ribosome, whether that's the ribosomal rRNA or the ribosomal proteins, which is what we tend to use these days. We tend to take all 53 ribosomal proteins as amino acid sequences, stick them all together on a computer to make an artificial, really long protein sequence of about 6,500 amino acid residues. And we use that because it's absolutely foolproof and gives really clean results. And then for the benefit of anybody who's just not got the vocabulary, the genome of an organism, because I'll be mentioning this quite a bit, is every gene an organism has in its DNA, not including small elements that can move between cells, but all the core genes an organism has. The gene itself is just a region of DNA that is the blueprint for a single protein molecule, a single polypeptide, or a single rRNA molecule. And when we write genes in bacteriology, it's different in other areas. Lowercase first letter, and we italicize it. And the protein that goes with it, we don't italicize and we capitalize the first letter. So you can tell what I'm talking about by the way it's written, essentially. And then the last one is operon, which is just a group of genes that are usually related in some way. They might be different subunits of a protein. There might be different proteins in a metabolic pathway. But when they're translated and transcribed, it all comes off in one piece of mRNA. That's the official definition of it, but you can just think of it as a group of genes that work together. So going back to the trees again, the numbers that you saw on the trees we've seen already, a lot of people get mistaken by those and they think they're something to do with percentage relatedness. They're just a statistical measure called a bootstrap value, which just tells us how reliable the data are, really. In short, the higher the number, the more reliable the tree topology is. Close to 100% is what we want to see. If they're less than 70%, we generally don't show them because they don't mean anything. And so the higher the number, the more reliable what you're looking at is. And I've already explained what these collapsed triangles are, just to show that you don't need to show every single organism name all the time. So the Thiotrichales itself currently is 6 families. It is an illegitimate name and does need to be dealt with. And we are in the process of dealing with that at the moment. And we've already done quite a bit of work on the Thiotrichales, which is the type family, and we split that, creating the Thiophyllaceae and the Thiolineaceae a few years ago now. We're currently working our way through the Piscivoricatiaceae, and that's what I'm going to be talking about today. So I'm really going to just talk about just this one family and really just a few genera within that family. So back in time a little bit, back before 2017, all the work that was being done in these groups of organisms, we all thought was one genus. We thought there was one genus, Thiomicrospira, and it was very big. And we got very different results with Thiomicrospira from all over the world. And that was a bit impeding to research because we were seeing differences that didn't really make a lot of sense. And there were 2 genera very close to it. There was Hydrogena vibrio, which only ever had 1 species in it. And there was Thioalkali microbium down here, which had 4 species in it at the bottom of the tree. And we looked at this and did quite a big taxonomic look at it in 2016, 2017. And this was work my group did in collaboration with Katie Scott's group in Florida, that we actually did it, got it as far as submission. And then she emailed me saying, oh, we really need to sort out that genus. It's a bit of a mess. And I said, well, we've actually just submitted the manuscript. So we unsubmitted it and then worked on it together for another 6 months and submitted a far better manuscript, but actually with some quite different results in the end. So we noted when we did that really it falls into 3 groups. And I won't go into how we use DNA relatedness measures to look at this, but that's what we were using. And I'll give a link to the paper at some point. So if people do want to see how we came up with the DNA relatedness measures to use, you can see that. We did have to define new ones because there were some issues. So what we did is decided there were 3 big clades here, and this is what we did. We took this top clade, and because that already contained an existing genus, Hydrogena vibrio, we couldn't create a new name, it had to take that name. So everything in there became Hydrogena vibrio, and this is going to become important as I'm going to show results on Hydrogena vibrio in a moment. And that annoyed a lot of people because that's a masculine name, so all the feminine endings had to become masculine endings, and people had to remember how to spell things again. Middle group we made into a new genus, and we spent a very long time coming up with a name that was similar, Thiomicrorhabdus. Rhabdus is from the Greek, so whilst it looks masculine, it's actually feminine, small sulfur wand, because they're all rod-shaped in this particular group. And because that's already feminine, we didn't have to change any of the endings of the names, which meant we could annoy fewer people, so that was important for us. And then lastly, this bottom group was a bit of an issue. The type species of Thiomicrospira is in bold here, Thiomicrospira pelophylla, and that had to stay because wherever the type species is, you can't get rid of that. That genus exists because of that species. Whereas this adjacent group, thioalkali microbium, turned out to actually all really be Thiomicrospira. Once we got rid of this upper group, these bottom two had to be merged into one genus. So all 4 species of thioalkali microbium became Thiomicrospira, and all the names changed from gender neuter to feminine because of the gender of the names. So that's where we got to, and that's how it now looks. And since we did that work, loads more species have been published, and actually several new genera have been published. So we've got new genera here in orange and red. Hydrothermal are all very neat at the top, Thiomicrorhabdus in blue in the middle, and Thiomicrospira at the bottom. And this was done using ribosomal protein concateners. So this is nearly 6,500 amino acid residues to make the tree. So it's extremely reliable. And I've not shown the bootstrap values on it because they're all 100%, because it's a very reliable tree. When you use a very, very long sequence, you get more reliable data because you're using a bigger sample effectively. And we've just this year published Thiomicrorhabdus hindhorstii and Thiomicrorhabdus cannonii. There are new ones that we've just published this year. But you can see the Hydrothermal group up at the top. Most of those are from the deep sea in the lower clade, and then in the upper clade, shallow sea, coastal seawater, that sort of thing. There's a few exceptions. There's the odd one from the salt lake, but the majority of them are from the deep seas. Thiomicrorhabdus in the middle is very mixture of deep seas and shallow seas. Some of them are in ankyline sinkholes where you've got saltwater and freshwater mixed together. And then Thiomicrospira at the bottom tend to be from very, very salty places or from clam gills, which didn't make an awful lot of sense to us at the time, but now it does as we've worked on this a bit more. So I'm going to first of all talk about a story that relates to carbon uptake by these organisms and how they cope with their environment. And to do that, unfortunately, I've got to explain a little bit of biochemistry, because when we're dealing with very small organisms, there is no behavior, there is no anatomy, there is nothing at organ level. Everything we're dealing with is at reaction level. So we have to kind of understand this to start with. Now, most of you will probably be familiar with the Calvin cycle, which is what plants use to take up carbon dioxide. And the business end of that cycle, if you like, is the enzyme rubisco, ribulose bisphosphate carboxylase oxygenase. In the bacteria, we have a version of rubisco, and actually we have lots of variants on it called isozymes or isoenzymes, which does the same job, has a different structure. And that allows them to do better at low carbon dioxide levels or at high carbon dioxide levels. And there is an inherent ropiness about this enzyme. It's not really a very good enzyme in terms of evolution, because it can do 2 different reactions, and one of them is good and one of them is bad. They both take the sugar phosphate ribulose 1,5-bisphosphate. First reaction, this is the one that we kind of want to happen, reacts it with carbon dioxide and produces 2 molecules of phosphoglycerate. All you need to know is phosphoglycerate's the stuff we want. The other reaction reacts it with oxygen. That only gives 1 molecule of phosphoglycerate and gives phosphoglycolate as well. Now, some plants can cope with phosphoglycolate. Plants have found a way to use it. Bacteria haven't. They secrete this into the medium, and actually we can use it to make glycolic acid for the skincare industry, as it's an important AHA for exfoliation. So we can exploit this reaction for biotech purposes. But the problem lots of organisms have in the wild is if there's not a lot of carbon dioxide around, this bottom reaction will happen all the time, and then they're going to struggle to grow and probably die. So evolution's found a way to deal with that, and that's something called the carboxysome. If you look at this electron micrograph at the top right, you can see about 5 big polyhedral bodies inside the cell. And you can't really see how many sides they are because of the resolution of the image. But they've usually got sort of geodesic dome sort of shape to them. And a carboxysome is simply a house for a couple of enzymes. It's got a protein shell on the outside. It doesn't have a membrane, so it's not an organelle. And inside, it's only got 2 things. It's got rubisco, which is the orange blobs, and a second enzyme, carbonic anhydrase, which under these conditions basically takes all the possible soluble versions of carbon dioxide, like bicarbonate and carbonate, and turns them into carbonic acid, which spontaneously decomposes into carbon dioxide. So what carbonic anhydrase is doing is ensuring any CO2 inside the carboxysome stays as CO2 gas and doesn't become bicarbonate or carbonate or anything like that. And the carboxysome lets in the substrates for the enzyme, and it lets the carbon dioxide in the form of bicarbonate, HCO3 2-, because at the pH of the cytoplasm, that happens spontaneously. It doesn't need to be converted. That's what carbon dioxide does at cytoplasm pH. Once it goes inside the carboxysome, it's turned back into carbon dioxide. So the interior of the carboxysome is always going to have really high carbon dioxide, very, very low oxygen, which means you get the great efficiency. You see, you don't then have a problem with the wrong reaction happening. And obviously that's evolved partly to deal with situations where you've got little carbon dioxide, lots of oxygen, but it's also used by organisms that just live in places where there's not a lot of carbon dioxide, because it gives them a way to concentrate it up. And if they can concentrate it up, they can get at it more easily. So I'm going to talk about some deep sea hydrothermal vent organisms and compare them with shallow sea hydrothermal vent organisms in the way they've solved this problem. And this really took us by great surprise. So going back to our tree, the two organisms we're going to be dealing with are Hydrogena crunogenus, specifically the XCL2 strain, just because it's been worked on a bit more, and Thyomicrorhabdus arctica, which is an Antarctic sediment isolate. So these are the two organisms, and if you look at the images at the top, you can see the left-hand one has got carboxysomes in it. They're not very clear, but you can see the little black particles inside the cell. A couple of them have got arrows on them to indicate that there are carboxysomes in it. And the right-hand image, Thyomicrorhabdus arctica, does not have any, and slices were taken through lots and lots of cells, and it never has carboxysomes. And we actually know that the genome doesn't have the ability to make carboxysomes. It hasn't got the operon. It looks like it's probably never had the operon. And if you want to read the full study on this, it should say Scott et al. It's got about 35 authors on this paper that we did, and you can read the whole story if you want to. So the bottom line is Hydrogena crunogenus is from a deep sea vent in the Pacific. It's about 2 kilometers down. It's under quite high pressure. It's at relatively temperate temperature, +2 to about +20 Celsius, and it's about pH 5.6 to 7.2, slightly acidic. And the total dissolved inorganic carbon, DIC, dissolved inorganic carbon is bicarbonate, carbonate, and any other forms of carbon dioxide in a dissolved state, regardless of what they are, lumped together. We just call them dissolved inorganic carbon. It's just convenient. Concentration is about 3 to about 7 millimolar, which is quite high. Thyomicrorhabdus arctica comes from a shallow sea sediment on the edge of a hydrothermal field. So it's not really from right next to a vent like the other organism is. So the water is a lot colder. It's between 0 and 6 roughly. And it's got lower dissolved inorganic carbon, and the pH is higher, and it's from relatively shallow depth, a couple of 100 meters really. And so we've got one on the left with carboxysomes, one on the right without carboxysomes. And that's very counterintuitive, because the organism that comes from the high dissolved inorganic carbon probably doesn't need carboxysomes, and yet it has them. And the one from low dissolved inorganic carbon logically should have them, but doesn't have them. So this was a great question as to why that was. So this started out when this great question started, that Katie Scott's group started working on it, and then partway through I got involved. So what they'd done initially was they looked at the growth of the two organisms, and they actually compared them with a whole load of other organisms in the interest of sort of fairness. And they grew them under the same conditions, and they actually grew them at different levels of carbon dioxide just to see what happened. And what they found out was this sort of thing. The scale on the y-axis is effectively a proxy for amount of biomass. So just think of it as amount of biological material formed. Hydrogena crunogenus is producing relatively low growth, but it does it quite quickly. Thyomicrorhabdus arctica does grow quite slowly, but it's producing much more growth. So it's certainly not struggling. It's doing better than all the other Thyomicrorhabdus. And some of those do have carboxysomes, probably because it doesn't need to make carboxysomes, which are very expensive to make. So probably can grow and produce more cells per unit carbon dioxide, we think. So it's certainly not struggling. It's certainly not a weak organism. In fact, if anything, Hydrogen Vibrio cruinigenus looks quite weak, and yet it's got carboxysomes, so doesn't make a lot of sense. So we refer back to the Bassbecking hypothesis, which in geomicrobiology is a very central hypothesis, which is in English, everything is everywhere but the environment selects. Originally, alles is overal, maar het milieu selecteert in Dutch. The first part of that, alles is overal, comes from Bayerink years before. Bassbecking extended it with the second part. Sometimes the literature cites the wrong person for it, but Bassbecking came up with this version of it. And of course, the environment does select. The properties of every organism on this planet are dictated by its environment, ultimately. So when you can't understand the physiology of the organism, what do? You look at the environment. Now, the trouble is with a deep sea vent organism, you can't just go out and collect a sample from 2,000 meters down years later and have a quick look and find out what the water is like and what the carbon dioxide concentrations are. A, technically very difficult. B, you need a submarine, that costs lots of money. And C, unlike soils, the oceans change constantly, which is why you can go to a field and you can find Roman relics that are still there after all this time, because the Earth's not moving around. But of course, the waters in the ocean are constantly moving and constantly changing. So that's not possible. But we knew the conditions it came from. We had some geochemical measurements from the original sample. So this is where I came in to do some modeling. So I ran some very, very standard geochemical models for the two different systems at a range of temperatures using these pressures. And we put into it the parameters of the seawater found in the two sites. So we used the standard seawater composition adjusted for the chemistry of Arctic sediment water versus hydrothermal vent water, which are quite different. And one thing that the astute might notice is that I've done a weird thing with the pH row. I've written the pH with the hydrothermal vent high to low, which seems a bit weird. The reason I've done that is just because all the values below it, the right-hand one of each pair relates to the low pH and the left-hand one relates to the high pH. So I did that just to make it not seem odd for anyone who does understand the data. And what I did is I modeled what happens at that temperature and that pressure to dissolved inorganic carbon total concentrations in that range or at that level with all the other things that are there, including hydrogen sulfide, including metals, including everything else, and looked at, well, what is the carbon actually in? What form is it actually in those conditions? What we found is in the Arctic sediment, there's very, very little carbon dioxide itself dissolved, which makes it even more counterintuitive. Because if there's no free carbon dioxide, it really needs carboxysomes. How is it coping? Whereas at the deep sea hydrothermal vent, there's a lot of carbon dioxide, and that's fine. There are other forms of dissolved inorganic carbon that I've not shown that are only present in tiny, tiny quantities, but these are the major ones. Bicarbonate accounts for most of what there is in both cases, and it's actually in a pretty similar ballpark. The total bicarbonate is roughly the same concentration across the two sites. So that kind of evens things out a bit. Some of these other species here, like sodium bicarbonate as an ion pair, or the magnesium bicarbonate and calcium bicarbonate ion pairs, are bioaccessible by the organism to a degree, but not brilliantly. Carbonate is very low because the pH is too low for carbonate really to appear. Bicarbonate seemed to be where the story was. But we found this very low carbon dioxide totally counterintuitive. It just made no sense. The hypothesis that we ultimately came up with is that because this organism grows really slowly, we figured that it never reaches a growth rate where it's actually struggling to take up carbon dioxide. It's growing so slowly that the bottleneck in its metabolism, if you like, is not carbon dioxide uptake. So that's probably one of the reasons why it just doesn't need carboxysomes. The other reason, which I'm only going to tell you just the tip of the iceberg of, because it's really Katie's work, and if you want to read it, it's in the paper, and it's very complicated genetics that I don't fully understand myself. Bottom line is that the organism on the right has got many, many, many more ways to bring bicarbonate ions into the cell. So it can raise its cytoplasmic carbon dioxide level dissolved in the cytoplasm, if you like, to really high levels just by transporting it into the cell much more quickly. So you can think of it having not just more doors that let the bicarbonate in, but doors that are specific for bicarbonate and are really good at letting it in versus the other organism. So it kind of gets round the need for traditional carbon dioxide concentration through a carboxysome by just having brilliant transport systems, and it can bring lots of stuff into the cell really quickly. And that really was a bit of a paradigm shift when we first found that. And it took us a long time to be confident enough in what we'd done, because that's such a big deal. And now other people have found the same sort of story, of course, elsewhere, as always happens. So my second sort of story I'm going to tell you is a bit more of a one closer to home, quite literally at the moment, because I'm in my office at work and I can see the isolation site where this organism came from right ahead of me at the moment. I didn't actually isolate it. It was an organism discovered in the 80s, just happens to be by coincidence right opposite where I now am sat. So two organisms I'm going to talk about now are true Thiomicrospira. Thiomicrospira pellophila, which is the type species of the genus, Pellophila mud-loving, or that which holds mud beloved. Thiomicrospira Thyasira, Thiomicrospira from Thyasira, which is a clam, specifically from the gill tissue thereof. So these two little organisms have been subject to a lot of debate since about 2005, because as you can see from the tree, there's very little difference between them. They're virtually identical in terms of this huge concatemer of ribosomal amino acid sequences. They're also near identical in ribosomal rRNA trees as well. So for a long time, there's been a debate of, are they separate species or not? And in part, that's been because they have been reported to have very, very different physiology, and that's always kept them separate. So Thiomicrospira pellophila came from a mudflat in the Wadensee. Thiomicrospira thyasira, yes, from the gill tissue of a clam, but of course that clam is living in shallow marine sediment, so relatively similar ecosystem. These various measures of diversity, it's virtually identical which either of these two measures you use. So we've been questioning, are they the same species? Are they separate species? What are they? And that's hence why Rory and Alex were working on this for quite a while to try and get an answer, which we now have. If you look at that genus more widely, you can see they come from incredibly similar ecosystems, same salinity, same pH, whereas all the others come from very high salinity ecosystems of as much as 14% salinity, very, very high pH, up to pH 10. These really are the two mild-mannered ones of the genus, and they have quite different morphology to the others, because what we've learned about this genus is they change shape as they grow. They start out as little rods like the top image, and then they gradually get curvy and spirally, and if you really stress them, they try and form a circle like this one at the bottom is trying to do. And that's just a stress response, and it's not that uncommon, but for a long time people believed to be in this genus you have to have this kind of spiral shape, so other things got rejected from it. And you can see here an example of all the different shapes in an image that Rory took on this image on the right, all the different shapes cells go into as they get older and when they're in different stages of division. And similarly in this image on the left, this very long cell in the middle is probably an older cell, and they can also form into quite long spirals. Everything on this screen is Thiomicrospira thyasirae at different ages of growth. So going to the clam, now bear in mind I'm not a clam biologist. The way these clams live is that they live just under the sediment, and they dig two sets of tunnels with their foot. They dig a tunnel above themselves, which is just a straight inhalant canal, which is only a few centimeters long. And then they dig a network below by stabbing their foot in and out of the sediment to dig a 20 centimeter long sort of tunnel with lots of side branches to give it more surface area. And that fills up with pore water from the sediment. And then what the clam does is it sucks water down from the bottom of the water of the sea, from water above the seabed, sucks water down, which is oxygenated water, And then after it's sucked water down, it sucks water up from the very sulfide-rich water in the sediment. And it does that about every 20 minutes. It swaps directions and goes back and forth. The reason it does that is it has mutualist symbionts, so that's a symbiosis where both organisms benefit, living in the gill tissue. And the hydrogen sulfide is used by those organisms at the expense of oxygen from the water above to make ATP and NADH, and then they fix carbon dioxide, which is found in the water. And as they do so, they cross-feed sugars and so on to the clam, to the point that 50 to 100% of the carbon in the clam's wet biomass, and I should be more precise about saying that, 50 to 100% of the dry biomass of the wet part of the clam, and 50 to 100% of the aragonite mineral in the shell itself is actually derived from bacterial uptake. And they can tell that based on the delta carbon 13 ratio, which is something I'm really not going to go into, but bottom line with it is carbon 13 should be about 1% of the total carbon on the planet. And sometimes it goes a little bit higher and sometimes goes a little tiny bit lower. And based on the pattern, you can predict which enzyme converted carbon dioxide into biomass ultimately. So they can predict whether something was biological fixation or geological fixation. Was it done by bacteria, or was it done by plants, or was it done by algae? And also which bacteria. The fingerprints are really very telling. So this has raised a question to us as to what Thiomicrospira thyasira is, because it is from inside gill tissue, but it's in the wrong place. It's not in the place where true mutualist symbionts of clams are absolutely universally found. And we know the true symbionts are Hydrogena species, which we've just met, and in some specimens, Magnetospirillum as well, which are a really, really cool genus. They've actually got a compass within them, so they can actually detect magnetic north and orient themselves accordingly, which is amazing biology. So we've had this great question as to not just are these two organisms the same species or not, but is one of them even a true mutualist symbiont, or is it something else? So one of the first things we looked at was based on the fact that the clam isolate, when you grow it in the lab, you don't need to give it vitamin B12. It can make its own vitamin B12 by itself. And if it's living in a clam, that means it could be feeding that vitamin B12 to the clam, because the only things that can make vitamin B12 are bacteria. We get all our vitamin B12 ultimately from gut bacteria, or from eating the tissue of animals that have taken up vitamin B12 from their own gut bacteria. We don't make it ourselves, plants don't make it, it all comes from bacteria. And Thiomicrospira pellophila, the mud-dwelling organism, can't make its own B12, so it must steal it from other cells in the sediment. Now, I knew from previous work I'd done with a related organism, Methylophaga, which is also in the Thiotrichales, that this is sometimes just an experimental artifact, because in the culture media that we use, which sort of mimics seawater, we have a habit of putting orthotungstate in the media. That's really because A, it's in seawater, and B, a very small number of organisms need it. These organisms don't need it, but we're creatures of habit. We tend to just always put it in the medium. And I found years ago that we always got an orange precipitate in these media. And some people would report Methylophaga needed B12, some would report they didn't. And eventually we figured out actually very few of them do. It's just a case that to make B12, you need cobalt. If you look at the diagram on the right, the middle of the B12 molecule is cobalt ion. And you can put ample cobalt in the medium, but the presence of tungstate removes it from the medium. So you can think you've given them enough, and actually they're starving and can't make it because you didn't give them enough. You give them some B12 and they'll use that instead and they'll be fine. So again, we did some modeling on this, and on the basis of how much we added to the medium, actually half of it was being turned into cobalt tungstate, orange precipitate, and only half of it was staying in some form that the organism could use. But that was a more than ample amount, and they should be able to use it. So we did a whole series of experiments where we removed the tungstate and we raised the cobalt concentrations to really high levels, and we could not get Thiomicrospira pellophila to grow without giving it actual B12. There was no way around that at all. And we tried all sorts of variations on that. Since then, we've now sequenced the whole genome of the organism, and we now know it's missing one of the genes. So at some point in its history of evolution, something in that lineage had it, because Thyamicrospira Thyasira has still got it, but Pelophylla lost it at some point along the line. And because it lives in a sediment where it can access B12 from other organisms, it survived, whereas Thyamicrospira Thyasira still makes it, and therefore must have evolved or diverged away before that event happened. Now, being able to make B12 is obviously advantageous to the clam because the clam can't make its own. So if Thyamicrospira Thyasira is feeding B12 to the clam, that could be evidence of a mutualist symbiosis. When we looked at these two organisms at genome level next to each other, I've kind of highlighted where the differences are. You can see the genome size is bigger in Thyamicrospira Thyasira, just in the size, number of base pairs, in the number of genes, and the number of protein coding genes. It is definitely bigger. Some of that difference is the presence of 1.5 prophage. A prophage is a sort of sleeping virus. It's viral DNA that's been incorporated into the bacterial genomic DNA. But if you add up the total of that, it's only about 1/3 of that difference in size. So it's not accounting for all of it. It has got genes and it has got things the other organism doesn't have. And when you look at these measures of diversity that work at whole genome level, these are measures like ANI, AF, and DNA-DNA hybridization done in silico rather than in vitro. Those measures compare the genome as a whole. So it compares all of the DNA of the organism with the other organism at whole genome level. It's not looking at one gene here, it's looking at every gene they have. And we have cutoffs for these are not the same species. And that is 95% for the first measure, 85% for the second, and 70% for the third. And you can see in all three cases, the difference is bigger than that. So these are most definitely different species. It's just that they've got very, very similar ribosomal rRNA and very, very similar ribosomal protein. And that tells us something. When we looked at where the big differences actually are, we went through the genome looking for things that were different. And it's in things like the sulfur oxidation pathways. It's in things like central metabolism. It's in things like the Calvin cycle. It's in things like the genes involved in attaching to surfaces, the things that probably relate to it attaching into the clam tissue, perhaps. Those are where the big differences are. Whereas the ribosomal rRNA and the ribosomal proteins are dotted all over the genome, and they evolve at a fairly constant rate across most but not all bacteria. The way they kind of evolve is fairly static, and so we use them as a measure of difference and a measure of speciation. The exception to that is in very, very fast evolving organisms like gut pathogens, where they have to evolve really quickly to cope with an immune system. You see very small changes in the ribosome and big changes in the rest of the cell. And you occasionally get the opposite, where you get big changes to the ribosome and very little change to the rest of the cell, usually in very, very, very slow growing organisms. In this case, this looked to us like probably a very recent speciation. So that difference is there, it's real, it's two different species, but it's happened incredibly recently. So we followed that up by taking the entire genome sequence of both organisms, and we actually took all the other Thyamicrospira at the same time, and we looked at them using an algorithm called time tree, which came from Marin et al. In 2017. So we couldn't do this work until really quite recently. And what this does is estimate when divergences happened between species based on the whole genome sequence, so every base of the entire DNA of that organism. It's not just looking at little fragments. And you can see that when Thyamicrospira really first emerged was about sometime around the point where the supercontinent Columbia was just starting to break up. And then we had another split happen about 230 million years ago in the Triassic. And then the other group, the group that live in soda lakes and so on, split again about 40 million years ago and 11 million years ago. But this split between these two organisms only happened 7,000 years ago, which is incredibly recent. Now, at that point, the environment was incredibly different, and I find it quite hard to envisage that 7,172 years before today, 7,100 before 1950, which is the value that the algorithm gives you, at that point, the British Isles were still connected by the Doggerland Plains to continental Europe. The world population was smaller than the population of the British Isles as they now are, and was virtually all in the Fertile Crescent. The ecosystem must have been so different at that point, and also so unspoilt. It's very, very hard to envisage what that would have been like. I mean, there wouldn't have even been a great pressure in terms of people harvesting the clams to eat them because the population was so small at that point. But this is just an incredibly recent speciation, but it's a real speciation and it's a real difference by all measures that we can use. Question we still have is this mutualism? Is this neutralism? Is this commensalism? What kind of symbiosis is this? Now, the zigzag of symbioses that I've shown on the right here, this is cut down. Really, there's another kind of zig, if you like, at the bottom, which is the things where there is a negative effect. So things like parasites, things like pathogens, they would be there. Now, we know that there's no pathogenic effect here because the clams aren't dying of infection. The clams are clearly benefiting in some, well, we don't know if the clams are benefiting from this particular organism, but they're certainly not benefiting. They're certainly not having a detrimental effect. So we've only considered the ones where there's a benefit or no effect. We've either got the situation where both organisms benefit, in which case it would be a mutualism, or one in which the host does not benefit, but the microbe does, which would be commensalism, or neither could benefit, and that would be a neutralism. Now, we can rule out neutralism straight away because Thiomicrospira is benefiting from that siphoning of water up and down all the time. That's what's feeding it. So it's benefiting. We know that. So either mutualism or it's commensalism. Now, the great thing about symbionts, and I'm not a symbiont biologist, but I've kind of fallen into this sort of backwards from working on other aspects of these organisms, are that the symbiont people have looked at genome features of mutualists and commensals and neutralists, and they've found all sorts of features that do and don't change that allow you to predict the relationship, which is really cool. So the percentage of G's and C's in the genome, generally, if you look at a true mutualist versus a close relative, the GC content of the genome, percentage of G's and C's in the genome as a whole versus A's and T's, is lower. The whole genome usually gets smaller, and the number of transpositions, that's just genes moving about, generally gets bigger. Now, what we see in Thiomicrospira Thyasira versus Pelophylla is the GC fraction is exactly the same. The genome is bigger, not smaller, by about 6% if you ignore the prophages, 10% if you include them. And it's got nearly 10 times more transposed genes than the mud-dwelling organisms. So that doesn't look likely to be a mutualist symbiont because it hasn't got the right features. It isn't evolving in the right direction, unless it's a very young relationship, of course. The other option is that it's a commensal. And the thing with commensals is they do tend to have prophages, more prophages than free-living organisms. Because of where they are in the gill tissue, they get exposed to more viral particles that are being siphoned up and down. And they tend to have CRISPR scars, which these organisms don't actually have, which are kind of signs of an old viral infection of the genome at some point in the past. So we have got evidence, yes, that it looks like it could well be a commensal, whereby the clam is not benefiting, but the microbe is. But it could also be a very young mutualism, where it is a true mutualist, just hasn't been one for long enough to have changed fully yet, and we're only seeing part of the story. So the next piece of work, of course, is to grow the organism with radioactive cobalt so that it makes radioactive vitamin B12 and find out if the clam tissue takes that up or not. And similarly, to see if there's carbon cross-feeding. But because of the location in the gill being so different from where the true mutualists are, it could be that no matter how much B12 the organism produces, the clam just can't get at it. So it's very likely this could be a commensal relationship rather than a mutual one. But that's really two areas of our work on the Thiotrichales to give you some idea of A, how we're doing taxonomy and systematics these days in the bacteria, which is increasingly becoming use the biggest piece of DNA you possibly can and ideally convert it into a protein sequence and use that, because it just gives you more data to work from effectively. Speaking to colleagues who are zoologists and botanists, they're having discussions about, oh, should we sequence this individual gene from all these organisms or not? Or should we pay for one genome and get a whole genome we can do something with? And they're almost about 20 years behind where we are now, because of course we're dealing with smaller genomes. We had genome sequencing before anyone else did, really, in our organisms. So it'll be interesting to see what direction that goes in the organisms with much bigger genomes. And genomes with a lot of redundancy, as that's one of the issues in plants and in reptiles particularly. The genomes get massive because they get filled up with multiple copies of things and with DNA that we don't really know the roles of. So the genomes get absolutely enormous to give them redundancy against damage effectively, whereas we're dealing with organisms with very efficient genomes that don't really have that. So it'll be interesting to see how using molecular taxonomy is going to evolve as a science in botany and zoology and the other areas of the eukarya over time, because obviously we're quite far ahead with that. But I think we're now reaching a point, because we're tending to use whole genomes, that we're going to start to see a lot more final decisions being made. When we were using a single gene, we knew it wasn't perfect. So every now and again, we've revisited with more genes, or we've used a region of the genome and we've kind of rejigged things. We found there were problems, we sorted them out, and they now seem to make sense. We checked them with the whole genome, they make sense. So I think we're reaching a point now where our use of molecular taxonomy, because we're always using the whole genome these days, we're not going to be seeing constant comp-novs happening over the years and constant reclassifications. I think we're going to be seeing things done much more robustly to start with, because we've got a better system now than we've ever had. And in a way, that's going to be awfully boring, because solving the mistakes that were made 30 years ago is always kind of fun in a way, and making order out of chaos. But we're going to be able to do things right the first time. And I think that is going to be really beneficial to the community over time. And hopefully I've shown you a bit of insight into the way these organisms evolve to suit their ecosystems in two very different sites, in marine vents and also in the relatively boring environment of the inside of a clam. And thank you all very much.
Thank you, Rich. That was absolutely fantastic. This is a field that is really far away from my own, and so I'll confess I didn't know much in advance of your talk in this area, and I have a ton of questions. But I also just didn't realize it was possible to really test those hypotheses about mutualism and symbiosis in such detail using these sorts of approaches. Fantastic. I'm very tempted to just ask you all of my own questions, but we actually have some questions from the audience. So we'll get through those first. And if there's time, I'll ask you mine, otherwise at a later date. So the first one is actually more about a comment. I know you addressed some of this, but maybe there's something more in here. Thiomicrospira Thyasira is not a symbiont. It was living in the marine sediment and it's actually outside of the gills when attempts were made to isolate the symbiont. I'm not sure that any symbiotic Thiomicrospira has been obtained in culture. Has anything ever been obtained in culture?
Not Thiomicrospira, Hydrogen and Vibrio, which were at one point Thiomicrospira, of course, until the last 3-4 years, 5 years now, they have been isolated. We have got Hydrogen and Vibrio, which are true mutualist symbionts of them. They've been isolated. Thiomicrospira Thyasira always had this debate of, is it real, is it not, or was it just attached to the gills? We've done some work with immunofluorescence, and we've actually shown that sometimes it is found inside the gill tissue, but it's not in the right place effectively. It's very shallow in the gill tissue, but it's consistent from clam to clam. It's almost always there. We've not really seen very many where it's not there to some degree. Now, whether that's just because it gets in and gets stuck, we don't know, which is why we believe it's probably more a commensal than a mutualist. But if it is a mutualist, it's very recent. But it's certainly a recent speciation in any case. Right. And I can kind of follow on because I can see Paul's other question. I did wonder whether Paul would come. So obviously Paul's diagram of the clam that I used earlier on from one of his papers. We have looked at some of the organisms lining the tunnel, and we've got a project student who's got a load of isolates at the moment, but we don't know what they are. The tunnel itself, because it benefits from that effect of siphoning up and down, kind of enriches a population of its own, as you would imagine would happen.
And the next question that we have in here, so let's pop this up, back up more. This is from Alan Hildrew, another Linnean Society trustee. Many aquatic invertebrates, including insects and amphipod crustaceans, rely on dietary algae for their essential supply of short-chain fatty acids. They can have them themselves. Bacteria and fungi lack them, hence the poor food quality of detritus compared with algae. Deep sea vents have no access to algal material. Do you know if these bacteria contain them, or perhaps vent animal species don't need them?
I don't actually know. When we look at fatty acids that are present in the membranes of these organisms, we don't really find anything below about 10 or 11 carbons in length. The really short chain ones, the kinds that you find in rumen fluid, the kinds you find in really stagnant soil and ponds, these organisms don't produce. In fact, they're actually toxic to a lot of these organisms. It could be, I mean, what you have to remember about vent sites is whilst you've got organisms doing primary productivity, whilst you've got organisms taking methane or carbon dioxide and making sugars and so on, those sugars leak out the cell and are eaten by other things. So even if these organisms aren't producing short chain fatty acids, the generalist organisms that live around the vents probably are. So somewhere in the chain, it might well be happening, but certainly in the membranes, these organisms don't have them. So they wouldn't come from the membranes of the dead cells, and they don't produce them as a side metabolite anywhere.
Thank you. Another question here from Avijit Datta. Great talk, especially on carboxysomes. We know that overall global oxygen and carbon dioxide levels have varied over time. Has this triggered relevant mutations? I had a very similar question. I'm very happy to see this one here.
If you want to know further about it, if you look at the work of Murray Badger in Australia, he's written a number of papers over the last about 20 years on all the different rubiscos and all the different ways carbon dioxide can get concentrated. And if you then map that back to when speciation events happen, you do see that there is some kind of congruency in the lines of descent that kind of imply that that's when they sort of happened. But what you do have to remember with the global oxygen and the global carbon dioxide is that's air. And most of the gas above stuff on the planet in places like caves or in the oceans or in bells of gas trapped in aquatic systems or even in soil is quite far removed from air because stuff's been taken out and stuff's been put into it. So what happens at global scale doesn't matter necessarily as much because microenvironments always exist. You've got 20% oxygen in air, but you only need 50 micrometers of dental plaque before you get zero oxygen because it consumes oxygen so much. So the global scale doesn't matter quite as much. But when we have looked at time trees and looked at evolution on a bigger picture of the Gammaproteobacteria, yeah, you do see there are points when certain groups of organisms appeared at the point when the carbon dioxide levels were really high or the oxygen levels were astronomical. Whether those organisms are still around, debatable. If they are, there's not very many of them left. What we see is a kind of common ancestor at that point, and then a load of evolutions happened since.
I guess a follow-up. You were talking about stable isotope ratios in terms of C13, C12 ratio, but that the uptake of those different isotopes in organisms is heavily dependent on temperature in many cases, right? With the heavier, the lighter isotope being taken up in different ratios during periods of global warming versus global cooling. Using that could affect your results.
I'm not sure what the impact is on it in the various different groups of bacteria and archaea that take up carbon dioxide, because we've got two different versions of the Calvin cycle with 7 or 8 different major isoenzymes of rubisco, all of which have got slightly different ratios to start with. And of course, they're only active in certain temperature ranges. So when that work is usually done, they do it at the optimum growth temperature of the organism to get standard values, and then those are then extrapolated away. But then there's all the organisms that don't use the Calvin cycle. There's the Arnon-Buchanan cycle, there's at least 4 others. I think we're up to 6 different pathways of autotrophy now that are known. So I don't think there are values for some of those organisms. Some of the other vent types, there are other types of vents that have different populations. There are vent types that have Guiparcharia. There are vent types that have epsilon proteobacteria. There are vent types that have other groups as dominant organisms. And some of those we are, like the archaea, we don't know all of their pathways of autotrophy yet, so we can't get values. and they're always sort of best estimates. But yeah, I mean, temperature undoubtedly has an effect, but whether it has an effect at, the temperature range within which that ecosystem actually ever is, it's not going up by 50 degrees. It's only got it might be high temperature or low temperature, but it's fairly stable.
Yeah, I was thinking about it more in the sense of, going obviously into the future where everything is kind of ticking up, whether that would affect some of these processes or not. But um, I see there's no other questions being asked yet. If anybody else has more questions, please do jump in. But if not, maybe I'll dive into the questions that I had, which were really about that. I'm not going to remember any of this species names here, but the two species that you're talking about in your kind of last section, the ones that diverged about 7,000 years ago. And until you'd actually said how long diverged they were, I was wondering how long diverged were these, because that's obviously such a striking difference, right, between them. But then looking at that time tree that you had, where it looked like there's that branch, and then the estimate for and I don't know if that's your full sample or if that's just the sample that you were showing in that slide, but the next kind of divergence estimate back is you're going into the billions of years, right, essentially, and then you had some others on the other branch were in the Mesozoic. And I was wondering how that sample might affect the calibrations, and what are you actually using for calibrating those points, since I guess I don't know what the fossil record really offers you in this field.
It's a really long-winded answer, but I'll give the bottom line. Basically, in that study that Marin et al. Did, they looked at organisms where there is a microfossil record and there's also genetic evidence, and they also worked back through various events and things as well, and they kind of came up with a calibration that works in the bacteria to give a kind of time tree. But the thing with molecular clock is it's always the best guess. I mean, it's not necessarily rigid. And of course, it's never linear. You can see in that tree that you've got something that's 7,000 years ago, and then the next branch to the left is 11 million years ago.
Yeah.
But those periods where those ancient divergences happened were periods where the atmosphere is very different or something big had happened. And in the actual algorithm itself, when it gives you its results, it actually maps it onto what the global CO2 was, what the sunlight levels were, had a meteor strike happened around that time, just so you can get a picture for what was the planet doing at that point. But as with everything that far back, we are ultimately guessing with everything in paleo anything. We're ultimately having to guess based on the best evidence we've got. And that's the best algorithm we've got at the moment, and that's what it's giving out. And it has been tested with really recent divergences in medical systems. So if you think about like a lung pathogen, it's not evolving to deal with like changes in metal concentration in the soil. It's dealing with an immune system, and an immune system adapts and an immune system evolves. So it's evolving against an adaptive system, not an evolving system, but an adapting system. So it's almost having to change against change. So you get really, really rapid speciation. You can get species forming really, really quickly, and it does pick those up. So it does pick up that those were in the last 100 years or whatever, or after the advent of penicillin and that sort of thing.
Which is extraordinary when you're thinking about a tree where you actually have nodes that are going back over a billion years. And in fact, it looked like those were the same genera in some cases across that. Is that right?
So that
was actually the same genera that were, that basically are going back hundreds of millions of years in that time tree.
It's one gene that in that particular one that I showed, that was one genus that was going back. It diverged away from the adjacent genus in some point around the time Columbia was breaking up. I mean, really that long ago. But what we think happened, if you look at the really big trees I showed right at the beginning, what we think happened with the bacteria is you saw a huge amount of diversification really early when cellular life first appeared, and then quite slow evolution, and then periods where it sped up because the selection pressures changed, and then it slowed down again. And one of my collaborators refers to it as sort of being like a maypole, in that you've got nothing happens for ages and then you've got stuff branching out, and then nothing happens for ages and then stuff branches out, because unless there is a selection pressure, why would anything change? You're going to get that kind of natural kind of bit of mutation that happens every X generations that just happens, but nothing's going to make a mutant survive more than whatever it mutated from unless the selection pressure changes, because something's got to change that makes the original one die out and the mutant proliferate. So unless something happens in those particular environments, you can have something stay the same for really long time. And when they pull things out of the ice record that have been there for huge periods of time, they're not that different from the organisms that we're dealing with today in some cases, because there wasn't a pressure.
So do you think that's different from calling it punctuated equilibrium?
No, I say that again. I didn't hear
Do you
it
think
properly.
that's different from punctuated equilibrium? Because to me, that sounds like a punctuated equilibrium model.
I don't know. I'm not a proper evolutionary biologist, so I don't know quite why some of these things happen. And I think also what we find increasingly is what works in ecological theory and evolutionary theory in things with arms and legs that walk about, and unicellular things where, you know, a millimeter is like miles for them. The scale that things are operating on, the models break down. And likewise, if you try and take E. coli's evolutionary model and apply it the other way, it doesn't work. So I don't think there ever will be a universal model. And that is in some ways why molecular clock never quite works. Because even across the bacteria, we know that there are different evolutionary models that apply in different areas. You do try doing a tree with one evolutionary model for the whole bacteria and you get nonsense. You have the different groups have evolved at different rates and they've got different levels of invariance. So whether ever an evolutionary model would ever apply to everything, I doubt it. And I think the bacteria are so diverse, and half of them we've never even seen. Half of them we've only ever detected sequences. We've never even seen them. I don't think we know enough yet. I don't think we have enough genomes to be able to answer those really big questions. But my feeling is that it's going to be different in different groups because of the rate of selection pressure.
I'm certain you're right that even it's not a lack of data, it's that biology is complex. Yeah. And we don't follow simple models.
Sadly.
Yeah. We've been working on morphological clocks recently, and if you want to make it even trickier than molecular clocks, there's no model. Well, I can sense that we are quite a bit over time, actually. But if anybody has any last questions, do feel free to pop them in. Otherwise, let me just thank Rich again for a really fascinating talk. That I love the introduction right at the beginning. Also, for people like me, that was very handy. But it really covered an incredible range of topics, and it was really excellent.